So there's several challenges with CAR T in solid tumors. One of them is targeting, which in this case the target is this marker guanocyclase C. It's a luminal marker in the colon, aberrantly expressed in colon cancer, and so we think we've addressed that to some degree by having a good target. The other challenges in solid tumor CAR T is not just hitting the target, but having enough target to promote expansion of the CAR and persistence of the CAR to generate an anti-cancer effect that persists and results in long-term control. And that's actually been, in some ways, one of the many holy grails of solid tumor CAR therapy, because you need to have a target to kind of bite onto, but you really don't want that target necessarily to be on normal tissue where there's co-expression of the target that's on the cancer. So by adding a CD19 CAR to this product, you achieve several things. First of all, we know the safety profile of CD19 CARs. This is very well established. There's really no questions. Well, there's many questions to be answered about CD19 CARs, but I think the safety profile of a CD19 CAR is established. We know that by infusing a patient with this bispecific CAR, it has the CD19 and the anti-GCC, that it will behave in a certain way. It will expand off of the peripheral B-cell population and the tissue-based B-cell population. It will promote growth of the CAR and it will promote survival the same way the commercially approved CD19 CARs are, but as the added target of the colon cancer target. So CD19 is a slingshot to launch the CAR, and it's fuel to maintain the CAR, and then the target is that guanocyclase C target. So the CD19 portion is a CD19 portion. It does the things you would expect, where there's CRS, grade 1, grade 2. There's neurotoxicity, ICANS, grade 1, grade 2, and they're managed in typical ways, and they're manageable and expected side effects. I don't think there's anything unexpected or unusual about that. The other observation in the abstract that we submitted and presented was that this causes an on-target effect of colitis. So keep in mind, GCC, the anti-colon cancer target, is also present on the normal colon. And so patients nearly universally develop some amount of diarrhea, and that's managed with steroids and anti-inflammatory drugs, things like infliximab or even vedolizumab, to tamp down the tissue targeting in normal tissue without overly suppressing the targeting in the malignant tissue. So at the time of this abstract, we were still in dose escalation. The trial is actually still in dose escalation. And so it's really early days. In the first dosing cohort, there was a response, and in the second dosing cohort, we're beginning to see responses. I think it's hard in dose escalation to comment accurately on disease control, but I would say there's promising signs of efficacy that you would expect in a well-targeted CAR therapy, starting at a low dose for safety purposes. The key thing right now is to find the recommended phase 2 dose and the toxicity management protocol for this CAR. And this is going to be the challenge for all the solid tumor CARs, because they're all going to be a little different. We're no longer just targeting terminally differentiated B cells like BCMA or CD19 or now CD22, for example. We're now talking about tissue markers, in the HER2 experience on the lungs and the CEA experience in the colon, and finding ways to tamp down too much activity that results in tissue injury without over-tamping that down, that results in lack of efficacy in the colon cancer in this case. And so we have a lot of work to do to find the ideal dose and the tox management plans, and as we get through that, we need to do those expansion cohorts that are planned in various populations to better understand who's who and what's safe. For example, in the GCC protocol, patients with an in situ primary are not permitted on the concern that a vigorous response could result in perforation. And we will eventually have to explore whether it's possible to give this to patients at risk of perforation. This clinical trial has a series of other sort of very strict criteria, and we have to, through cohort expansions, liberalize those criteria and see really who's the ideal phase 2 population.